Hello, my name is Sam Zakhari, and I would like to chat with you today about cardiovascular effects of ethanol and organ damage. But before we start, I would like to briefly discuss some background. Ethanol is the chemical name for alcohol, or ethyl alcohol, or grain alcohol, drinking alcohol, or simply alcohol. When we talk about alcohol drinking, we mean ethanol drinking. Ethanol is formed mainly by the fermentation of sugars and other carbohydrates by yeast. It is the active ingredient in alcoholic drinks, whether it is beer, wine, or spirit. On the left, you see the chemical structure of ethanol, CH3, CH2OH. And the figure below shows the spatial configuration of the molecule. I would like to call your attention to a lower homologue of ethanol, which is called methanol, which has one less CH2 in its structure. And the reason I'm telling you that is that methanol is invariably used to adulterate and fortify homemade illicit alcohol beverages. Methanol is highly toxic and it can be fatal because it causes CNS depression, central nervous system depression, and it's also metabolized to formic acid, which can cause blindness if the person is lucky enough not to die. All alcoholic beverages contain the same ethanol molecule, and that's the title of this slide, alcohol is alcohol is alcohol. However, the ethanol concentration varies between various types of drinks. For example, beer usually has 5% alcohol, although some craft beers go much higher than that. Wine is usually 12%, and it can go higher. And the spirits usually about 40% or even slightly higher. That said, the amount of alcohol in a regular drink or what's called equivalent drink is the same, which is about 14 grams or 0.6 fluid ounces of ethanol. The volume of a regular drink or a standard drink varies. For example, beer is 12 ounces, wine 5 ounces, and a shot of spirit is 1.5 fluid ounces. And this arrow shows the level of 5 ounces in a glass of wine. Usually it is half of the glass. Now, alcohol's effect on health depends on blood alcohol concentration, or what we call BAC, which is determined by several factors, mainly the patterns of drinking here, but with other factors as well. And sipping, as you know, sipping a glass of wine with dinner will not appreciably elevate blood alcohol concentration, but binge drinking will result in high BAC. So let me discuss the pattern of drinking. The first one is moderate drinking, and the dietary guidelines since its inception and including the 2020 to 2025 version have defined moderate drinking as no more than 1 drink per day for a woman and no more than 2 drinks per day for a man. The second pattern of drinking is called the high risk drinking, which is associated with harm both to health and society. 2 patterns of drinking fall under high risk drinking. Heavy drinking, when a person drinks too much too often, usually 4 or 5 drinks in a short time and repeated often. And the second one is binge drinking, which is drinking too much too fast. Binge drinking is defined as drinking 5 plus drinks for a man and 4 plus drinks for a woman in about 2 hours. This pattern of drinking will result in a blood alcohol concentration higher than 0.08%, which is the legal limit of drunk driving. Several factors influence the blood alcohol concentration, or BAC. BAC is determined by alcohol rate of absorption here. into the blood and the rate of its elimination from the blood. Other factors influence BACs such as drinking on an empty stomach, body weight, gender, etc. I would like to call your attention to what's called auto-brewery syndrome, which occurs in some people. And those individuals have a high BAC without drinking because their intestine ferment ingested carbohydrates to form ethanol. People with auto-brewery syndrome should not consume carbohydrates. Problem solved. Elimination of alcohol occurs mainly by metabolism in the liver and by its excretion unchanged in the lungs, urine, sweat. Alcohol is eliminated at a rate of about 1 drink an hour or an hour and a half. Now, we will discuss the effects of alcohol on the heart, and alcohol effects on the cardiovascular system is determined by the amount consumed. Like Janus Head, alcohol effects has two phases. At low to moderate concentration of drinking, alcohol is associated with improvement in cardiac function and vascular health, whereas chronic heavy consumption of large amounts of ethanol results in hypertension and cardiomyopathy. So, moderate drinking has been shown by numerous epidemiological and biochemical studies to decrease the risk of coronary artery disease. The effect of alcohol consumption on the risk of coronary artery disease is usually referred to as the J-shaped curve, which means if we assign a risk of 1 to people who do not drink, moderate drinking reduces the relative risk below 1, but heavy drinking increases the relative risk to higher than 1. The J-shaped curve has been identified by epidemiological studies. However, back in the 80s, several scientists, including Professor Shaper from England, claimed that the control group of non-drinkers includes ex-drinkers, what he referred to as sick quitters, who stopped drinking for health reasons. And that could exaggerate the coronary artery risk in non-drinkers and the benefits of moderate drinking. However, numerous studies repeated their analysis by removing the sick quitters and using data only from abstainers and found the same J-shaped correlation with decreased risk of coronary artery disease with moderate drinking and increased risk with heavy drinking. So now let us talk about what is coronary artery disease. Coronary artery disease, also known as atherosclerosis, is a pathological process that ultimately results in impeding blood flow in the coronary arteries, causing heart attacks that may culminate in death. Atherosclerosis starts with endothelial dysfunction, resulting in infiltration by monocytes, which then mature to form macrophages that phagocytose the droplet of fat from oxidized LDL, resulting in the formation of what's called foam cells, causing narrowing of the coronary artery lumen, as you see here. Macrophages play an important role in atherogenesis by participating in the uptake and metabolism of lipoproteins, secretion of growth factors, and production of enzymes and toxic metabolites that may facilitate plaque growth. Plaques with increased lipid content appear more prone to rupture, eventually forming blood clots. Here you can see clotted blood that prevents the blood flow and hence oxygen to the heart, resulting in heart attack. The extent of the resulting damage to the heart depends on the size of the clotted artery. If it is here, up higher, or here, that will affect the area that is infarct. The mechanism by which alcohol decreases the risk of coronary artery disease are plenty, and involve mainly decreasing blood clotting. However, I will enumerate other mechanisms without going into much detail. Moderate drinking increases serum tissue plasminogen activator, increases HDL, serum HDL, that's the high density lipoprotein, the good cholesterol, and increases adiponectin, and insulin sensitivity, which reduces the plasma glucose level and resulting in endothelial protection and enhancement of cardiovascular health, especially in diabetics. In addition, moderate drinking reduces the stress-related brain signals, which reduces blood pressure, oxidative stress, and blood clotting, as I mentioned earlier, all of which contribute to decreased risk of cardiovascular disease. Now, this slide shows the autonomic nervous system, ANS, how it controls the heart. The sympathetic and parasympathetic divisions of the autonomic nervous system innervate cardiac muscle and mediate a variety of reflexes that maintain cardiac homeostasis. The sympathetic nervous system increases heart rate, cardiac output, myocardial contractility, and blood pressure, while parasympathetic nervous system slows the heart rate. Activation of the arterial baroreceptors and the aorta and carotid sinus signal a rise in blood pressure to the nucleus tractor solitarius, which ultimately result in an inhibition of the vasomotor tone and excitation of the parasympathetic neurons. This results in a fall of both arterial blood pressure and heart rate, known as baroreceptor reflex. Alcohol consumption attenuates the hemodynamic response to baroreceptors activation by selective blockade of the NMDA receptors within the nucleus tractus solitarius. In this slide, I would like to list associations of moderate drinking with potential health effects based on epidemiological studies. Some of them, like coronary artery disease, ischemic stroke, and type 2 diabetes, are also supported by biochemical studies. I just want to remind you what is moderate drinking. It is no more than 1 drink per day for a woman and no more than 2 drinks per day for a man. It is important that no one should be encouraged to drink alcohol to achieve any health benefits. On this slide, I will discuss the long-term health effects of excessive drinking, and excessive drinking was defined in slide number 6. Long-term excessive drinking can exacerbate comorbid conditions, causes addiction, liver disease, including cirrhosis of the liver and cancer. And chronic heavy drinking also suppresses the immune system, causing muscle wasting, osteoporosis, pancreatitis, cardiomyopathy, and hypertension. And in the brain, chronic heavy alcohol consumption causes structural and functional damage, disturbed sleep, and hemorrhagic stroke. Also, epidemiological studies reported association between alcohol consumption and different cancers. Let me just have a brief discussion about hemorrhagic stroke. Increase in intravascular pressure exploits weakness in the arterial wall, whether it is due to congenital or atherosclerosis, hypertension, bacterial infection, or trauma, which causes aneurysm. Hemorrhagic stroke is due to the occurrence of a pinhole in the wall of the blood vessel that nourishes the brain, resulting in seeping of the blood, which comes in direct contact with the brain tissue, causing excessive oxidative stress and damage to the brain tissue. Hemorrhagic strokes represent only 20% of the total number of strokes, And the remaining 80% is ischemic stroke, which is due to narrowing of the blood vessels or clogging of the blood vessel, as we mentioned before. Now, chronic excessive alcohol consumption can result in cardiomyopathy in certain individuals. So what's cardiomyopathy? Alcoholic cardiomyopathy is heart disease caused by long-term alcohol abuse, which weakens and thins the heart muscle, affecting its ability to pump blood. This can lead to heart failure and other life-threatening health problems. In this picture, you can see the difference between the size of a normal heart and a heart with cardiomyopathy. The only treatment for cardiomyopathy is to excise part of the heart to reduce it to normal size. So, how alcoholic cardiomyopathy happened? Direct effects of alcohol, including increase in mitochondrial dysfunction, oxidative stress, heart size, referred to as hypertrophy, and decrease in protein synthesis. Those effects cause decrease in contractility and the ejection fraction. And the ejection fraction is the amount or the percentage of the blood ejected from the heart by each contraction. The second effect is indirect effect due to increases in sympathetic nervous system activity, renin-angiotensin aldosterone system, increase in the activity of that as well, and endothelial dysfunction. And this results in fibrosis and hypertension. Now, let me discuss briefly alcohol metabolism. How does the body metabolize alcohol and what are the consequences? Basically, there are three ways that the body gets rid of alcohol. The most important one is the oxidative pathway in the liver, which removes about 90% of alcohol. Less than 10% is excreted as unmetabolized alcohol in breath, urine, and sweat, as I mentioned earlier. But non-oxidative pathways and conjugation to glucuronic acid or sulfate also removes a small portion of consumed alcohol. Oxidative metabolism results in the formation of acetaldehyde and acetate and reactive oxygen species, increase in the NADH to NAD ratio, thus affecting the redox state of cells and derangement of metabolic pathways. And also chronic alcohol consumption results in CYP2E1 induction. Now, let me discuss the most important pathway of alcohol metabolism in the liver, and that's the oxidative pathway, which happens in hepatocytes or liver cells. First, alcohol is metabolized to acetaldehyde by alcohol dehydrogenase, ADH, in the cytosol, and to a lesser extent by CYP2E1 in the endoplasmic reticulum. A small amount of acetaldehyde is consequently metabolized to acetate by ALDH1 in the cytosol, but the vast majority is metabolized in the mitochondria by ALDH2 to acetate. The capacity of ALDH to remove acetaldehyde far exceeds that of acetaldehyde generation by the various pathways of alcohol oxidation. As a result, under normal conditions, circulating levels of acetaldehyde are usually very low. Much of the acetate produced leaves the liver and is activated to acetyl-CoA in extrahepatic tissues to produce ATP, which is used as energy source. So, to summarize, the effects of alcohol oxidation result in acetaldehyde formation, acetate formation, reactive oxygen species formation, and increase in NADH to NAD ratio due to alcohol metabolism by CYP2E1 and reoxidation of NADH and the mitochondria. This will also result in changes in important metabolic pathways such as retinol, tryptophan, etc. Now, let me discuss what's called polymorphism in alcohol metabolism. As I mentioned earlier, alcohol dehydrogenase metabolizes alcohol to acetaldehyde, which is then metabolized by aldehyde dehydrogenase to form acetate. Now, there are genetic variations or polymorphisms that exist in various ethnic groups, resulting in some enzyme variant that work more efficiently than others, suggesting that people can metabolize alcohol at different rates, resulting in a wide spectrum of health effects. For example, Asians, about 50%, are with defective ALDH2 enzyme that we refer to as ALDH22, and would have higher acetaldehyde level, resulting in accumulation of acetaldehyde that results in flushing and other health effects. Now, certain compounds called alders, you see here, that were discovered by an NIAAA grantee can activate the inefficient ALDH2 to metabolize acetaldehyde. There are also variations in the ADH enzyme. They are listed here in the yellow box. Some of them are slow metabolizer, and some of them are fast metabolizer for alcohol. And that, of course, affects the rate of acetaldehyde formation. The fast alcohol metabolizing enzyme, ADH1B2, is more prevalent in Jewish populations. Now, as I mentioned earlier, the results of alcohol metabolism, first one is the formation of acetaldehyde, which is very quickly and very efficiently removed by aldehyde dehydrogenase. However, any small amount that accumulate of the reactive acetaldehyde can form adducts with proteins, resulting in enzymatic inactivation, decreased DNA repair, and cytotoxicity. It also forms adducts with liver microsomal proteins, hemoglobin, and serum albumin and lipoprotein. As I told you in slide 19, acetate formed from alcohol metabolism is used as energy source in extrahepatic tissues. In the following 3 slides, I will discuss how acetate is used as energy source in preference of glucose, for example, in brain tissue, and how acetate exerts its own effects on the brain. Now, in this slide, acetyl-CoA is the unit of energy in both the mitochondria and the cytosol to form ATP. Now, just to make sure that you remember that acetyl-CoA is the unit of energy, I put acetyl-CoA here in the unit of monetary unit in the US, which is the dollar, and this is acetyl-CoA, and it is the unit of energy in both mitochondria and cytosol. Now, in the mitochondria, acetyl-CoA is formed from 3 sources. Number 1, glucose through glycolysis to pyruvate. Number 2, beta oxidations of fatty acids. And number 3, acetylated amino acids like acetyl-L-carnitine. All of these form acetyl-CoA, which is used by the mitochondria to produce energy. In the cytosol, however, acetyl-CoA is mainly formed from 2 sources. 1, citrate, which is extruded by a carrier from the mitochondria into the cytosol, and 2, acetate from alcohol metabolism and other sources that can be transformed to acetyl-CoA under the influence of acetyl-CoA synthase. Acetyl-CoA in the cytosol is used for fatty acid synthesis and also for formation of phospholipids and cholesterol. So this is how acetate is used as a source of energy. Now, alcohol is metabolized also in the brain, but for several decades, the prevailing dogma was that catalase is the main source of metabolizing alcohol in the brain, and to some extent, CYP2A1. However, in 2013, Wang and his colleagues demonstrated that astrocytes can metabolize alcohol, and in 2021, very recently, scientists from NIAAA showed that acetate is produced by aldehyde dehydrogenase in astrocytes. And acetate in the brain is used by aldehyde dehydrogenase also and is used as energy source, and it is used to acetylate histone and influence growth and cell proliferation, and is also used in cell signaling. This slide shows that acetate is significantly utilized by astroglia in preference of glucose. As well, lactate and ketone can be transported in the brain and used instead of glucose. In 2013, Jiang and colleagues demonstrated that acetate is taken by astroglia in the brain of heavy and light drinkers by injecting carbon-labeled acetate, resulting in the enrichment of glutamate and glutamine in the brain. Now, acetate also is metabolized to acetyl-CoA, as I mentioned earlier, and this results in the increased concentration of adenosine monophosphate, which is further metabolized to form adenosine. And adenosine has its own pharmacological effects. It is a vasodilator. It causes ataxia or sleep, depression of the central nervous system, and alcohol reinforcement. Now, the third result of alcohol metabolism, as I mentioned earlier, is the formation of reactive oxygen species, which results in lipid oxidation and oxidative stress, and which results in oxidative stress in the cell. Now, the sources of reactive oxygen species are two, as I mentioned earlier. One, alcohol metabolism by CYP2A1, and second, the reoxidation of NADH in the electron transport chain to form reactive oxygen species. It is important to note that some reactive oxygen species is essential for promotion of immune defense, so it is not bad at all, small amount. However, in large amount, it can cause tissue damage. Reactive oxygen species also can induce epigenetic effects, resulting into tissue damage. Now, reactive oxygen species, here is examples of the reactive oxygen species, and they are mainly chemical species that are active. And the most important one is the superoxide anion, this one, which is formed from oxygen by the electron transport chain or by CYP2A1. Now, superoxide radical is very unstable and interacts with nitrogen oxide to form peroxynitrite here. And the enzyme manganese superoxide dismutase reduces superoxide radical to form hydrogen peroxide H2O2, which in turn can be reduced by iron to form a nasty compound hydroxyl radical, or detoxified by catalase to produce water and oxygen. Hydrogen peroxide radicals can damage surrounding macromolecules, lipid, proteins, and DNA, causing mitochondrial dysfunction, causes epigenetic effects, and activates transcription factors that result in formation of inflammatory cytokines, resulting in tissue damage. The hydroxyl radical causes lipid peroxidation, which results in tissue injury. Now, the fourth result of alcohol metabolism is the increase in hepatic NADH to NAD ratio. And that causes derangement of certain metabolic pathways with consequential health effects. Although there are numerous examples of this derangement, I am going to discuss the results of the increase in NADH to NAD ratio. It results on formation of ketoacidosis, modulation of gene expression, especially SIRT1, and inhibition of a variety of biochemical reactions listed here. Now, the fifth result of alcohol metabolism is the derangement of certain metabolic pathways with consequential health effects. Although there are numerous examples of this derangement, I'm going to discuss only retinoic acid homeostasis. Now, as you know, alcohol dehydrogenase and aldehyde dehydrogenase metabolize alcohol to acetaldehyde and acetate respectively, as well as numerous other compounds, including retinol to retinaldehyde and to retinoic acid as well. Now, because both ethanol and retinol are substrates for ADH, consumption of ethanol will act as a competitive inhibitor of ADH-catalyzed retinol oxidation, and this may result in a reduction of retinoic acid synthesis and the consequent alteration in gene regulation via retinoid signaling pathways. Retinoic acid is used as a transcription regulator of key genes during growth and development. And this derangement of retinoic acid synthesis by alcohol could be a factor in the genesis of fetal alcohol syndrome. Now, the last thing I want to discuss with you is that alcohol induces cytochrome P450-2E1, which is known as CYP2E1 as well. CYP2E1 is a minor pathway, as I mentioned earlier, of alcohol metabolism, and it is induced not only by alcohol, but also by acetone, toluene, and other compounds. So what are the consequences of CYP2E1 inhibition by alcohol? It affects, of course, the activation of pro-carcinogen like the azo compounds and the nitroso compounds, pro-carcinogens. It can form reactive oxygen species from medications such as acetaminophen, resulting in hepatotoxicity. And to summarize the consequences of CYP2A1 induction, here they are. One is the formation of acetaldehyde and 1-hydroxyethyl radical, activation of molecular oxygen and reactive oxygen species, superoxide radical, as I mentioned earlier, activation of drugs and chemicals such as acetaminophen and halothane and benzene, and activation of pro-carcinogens such as nitrosamine, azo compounds to active carcinogens. Now, the next two slides summarize minor ways of alcohol removal. One of them is the metabolism of ethanol through fatty acid esters by fatty acid ethyl ester synthase. And the second one is the change of ethanol by phospholipase D to form phosphatidyl ethanol, which interferes with the phospholipase D dependent signaling. The last form of alcohol removal is not by metabolism, but by conjugation. Alcohol is conjugated with glucuronic acid to form ethyl glucuronide, and is conjugated also to sulfate to form ethyl sulfate. Although this is a minor way of removing alcohol, those products, ethyl glucuronide and ethyl sulfate, are used as biomarkers for alcohol consumption. So the take-home message that I would like to leave you with is that, one, low to moderate ethanol intake is associated with improvements in cardiac function and vascular health. Chronic consumption of large amounts of ethanol is toxic to the heart and can result in hypertension and cardiomyopathy. Oxidative pathways of alcohol metabolism produce acetaldehyde and acetate that play major roles in health effects. And finally, there are multiple genetic variations in alcohol metabolizing enzymes that can contribute to health outcomes. And with that, I will stop here. Thank you for listening.